Hi, welcome back. While last episode was all happy fun times discussing the awesomeness that is our replication system, there is still a lot of problems to discuss with regards to retaining your genetic code. In this episode, we'll tackle a couple of these issues and then ramp up from simple fixes to the most daunting and elaborate ones. So, let's start as simple as possible. And by simple, I mean throwing out these oversimplified nucleotides in favor of something which resembles a molecule. That's right, it's chemistry time. Even your DNA is regularly subject to completely random chemical processes which can change nucleotides on a whim. Here are some common examples. Water can remove amino groups in a process called deamination. A methyl group can attach itself to some spots on the nucleotide. Nucleotides can also just be whisked away, off to fulfill their dreams, I imagine. And oxygen can also cause a variety of destructive behavior. There are many more examples that fall under one of these four groups. All of them are examples of DNA lesions, which is a term that describes anything atypical on the DNA that isn't supposed to be there. The lesions that I just discussed fall under the category of chemically random or stochastic lesions, meaning that they can happen anywhere, anytime, with bare minimum outside influence. The biggest consequence for these is that it's possible for the wrong nucleotide to pair up once the replication cycle kicks in, resulting in a permanent mutation by the second cycle. As a result, these lesions have to be fixed, preferably before replication starts. But that's not all. Let's talk about some of the more destructive lesions. There are many cancer-inducing substances that exist in this world, not just in labs, and they can do many harmful things. For instance, chemically modified versions of nucleotides foreign to your body can enter your DNA and bind to the wrong conjugate pair during the next replication cycle. There are substances that can get between base pairs, making it hard to separate the two strands, causing major blockages for systems that rely on that mechanism. Molecules called bulky adducts like aflatoxin B1 can also seriously throw off DNA structure and regulation. Trouble can also come from outside as well. UV rays can bind two bases on the same strand, and even more potent nuclear radiation can cause DNA strand breaks, the most terrible of all lesions. So, this all sounds bad. And if all these lesions were to stick, it would be terrible. In fact, it is estimated that just 3 extremely well-placed mutations can have some disastrous results. To show you just one of these, here is a gene that encodes for a protein called HRAS oncogene. The name kind of already gives it away. This protein needs GTP to be turned on. Once it has encountered some GTP, it will switch configuration and is now capable of signaling that the cell needs to divide itself. If this were normal, wild-type HRAS, then at some point one of the phosphates of GTP would drop off, leaving GDP, which returns RAS to an off state. However, if the 34th base on the encoding part of the gene got replaced with thymine, the chance of GTP losing a phosphate lowers drastically. HRAS will continue to signal the cell that it should divide, and sure, there are mechanisms that counter this. But 2 more point mutations like this, and those mechanisms are gone, and we are dealing with a cell that is defined as cancerous. This is the cause for the most common type of cancer there is. So, at this point in the video, you might be a bit scared. But don't be. Because for the most part, where there is a problem, evolution has found solutions. Our bodies are powerhouses of DNA regulation, both recognizing and fixing tons of mistakes made. This is what the rest of this video and the next one are going to be about. There is a long list of defense mechanisms to get through, so let's tackle these problems. The simplest type of damage reversal is called direct damage reversal, where there is one protein that recognizes a mistake and immediately fixes it. This method is fast, efficient, and the least error-prone of all solutions that we're going to talk about. But guess how many of these proteins we mammals have? It's just one. For one type of lesion, 6-oxomethylated guanine can get fixed by methyltransferase. It's disappointing, right? Even bacteria have more proteins like this. They can fix these cross-nucleotide bonds created by UV light with one protein. In mammals, this mechanism has most likely evolved into our circadian rhythm. So, let's move on. For most lesions, the general solution is DNA excision repair, of which there are two types. One is a deliberate cut out one base and quickly repair it method. The other is a less subtle rip an entire section open and start again method. Let's take a look at the first, base excision repair. A protein of the family glycosylase recognizes a DNA lesion specific to that type of glycosylase. This glycosylase then turns the nucleotide around and removes the base. An endonuclease, in this case APE1, will then cleave the bond at the backbone. This leaves a gap open that other, less important proteins notice, which attracts polymerase. This polymerase then places a new correct nucleotide, and then the final gap is sealed by ligase. At this point, the reparation process is finished. This process fixes most of the changes done to nucleotides by stochastic processes like methylation, certain oxidations, and deaminations. It is also very good at recognizing and repairing naturally occurring single-strand breaks and missing nucleotides. What it is not good at, however, are removing some heavier substitutions. These are usually too big for a base excision repair, or they are limiting in some other structural way. These intrastrand products, for instance, bridge the gap between strands, and there is no glycosylase that is able to deal with that. And so now we have to enter the other excision repair method, nucleotide excision repair. Side note, due to both the difficulty of finding X-ray crystallography studies on some of the upcoming proteins and their sheer size, the following explanation will be done by slightly more cartoonish representations than my standard for quality would normally allow. If that's okay, let's move on. The biggest difference between base and nucleotide excision repair is the initiation process. In contrast to many different glycosylases, recognition of these bigger, more destructive DNA lesions is done by only one protein complex, of which the main part is called XPC. Now, brace yourself. Lots of proteins coming your way. XPC recruits a large protein complex called TF2H, which bind opposite to XPC at the site of the DNA lesion. Two subproteins of TF2H called XPB and XPD act as helicases and unwind the strand on both sides. This leaves two unwound single strands of DNA, and that is very dangerous, so on the side of the lesion, XPA stabilizes the strand, and on the other side, a single strand binding protein called RPA stabilizes the strand. At this point, the complex is in place, and it is stable enough to let XPC know that it has done its job and that it can leave now. After that, it recruits two endonucleases called XPG and XPF to put two cuts into the damaged strand. TF2H will then leave the strand with the excised damaged DNA, and after that, it's just a matter of filling the gap with polymerase and finishing the job with ligase. This system is able to remove a lot of lesions that can't be fixed by base excision repair due to the decrease in specificity. This decrease is because XPC doesn't bind to lesions per se, but to places in DNA where the structure is not as stable. There are a couple of caveats, however. If XPC binds to the site of the lesion, TF2H will still come to check it out, but it will quickly dissociate once the helicases aren't able to find anything. XPC will also not bind to any lesions that don't disturb the helix. This is still quite problematic because there is still one group of lesions not accounted for in mammals, and if you remember back at the start, UV-damaged DNA is still not fixed because we lost that direct reversal mechanism. To compensate, a protein called UVDDB has a high affinity for these UV products, and it is able to call XPC over to convince it that there is definitely something wrong here, even if XPC doesn't initially notice. And from here it is your standard cut open, snap out, and patch up that should become familiar to you. Join me next time for even more complex machinery when we discuss DNA double strand breaks.